feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinar. Thank you for joining us. Today we will talk about central and peripheral innervation. Professor Felipe Court is with us tonight. Please use the Q&A function to send your questions, comments. All your suggestions are very welcome. As always, we are starting with a short presentation, this time by Grace Kay, our research assistant at the NUHS Center for Healthy Longevity. And she will talk about the TFEB activators as potential geroprotectors.
Thank you for the introduction. As mentioned, I'm Grace, a research assistant under Dr. Esther Wong's lab. So today, I'm going to talk about my project, which involves high-content drug screening for novel TFEB activators as potential geroprotectors. To delay aging, we must first target the fundamental aging pathways. So looking at the hallmarks of aging, targeting the primary hallmarks colored in blue might limit the initial damage that contribute directly to the aging process. Hence, we decided to target the age-related decline in macroautophagy activity for this project. But what exactly is autophagy? Autophagy is a combination of two Greek words which translates to self-eating. It is a self-renewal process that keeps the cellular environment clean by recycling waste material via the lysosomes. So initially, a double membrane vesicle surrounds the unwanted material. During this process, LC3, which is represented by these green dots, is added onto the membrane. These vesicles then fuse with the lysosome, where enzymes break down the contents inside. Simply put, these blue vesicles are like a rubbish truck to transport unwanted material to the purple lysosome, which acts as an incinerator to degrade them. And this is where TFEB comes in. TFEB tackles two arms of the autophagic pathway by increasing the formation of both the autophagosomal vesicles and lysosomes. So with more rubbish trucks and incinerators, autophagy activity is increased. But what exactly is TFEB? TFEB stands for transcription factor EB. When TFEB is activated, it travels from the cytoplasm into the nucleus. TFEB can then act as a switch to turn on genes in the clear network. So this means that TFEB has the power to increase the formation of both vesicles and lysosomes, as well as improve lysosomal function. Hence, TFEB is known as the master regulator of autophagy and lysosomal biogenesis. By tackling two arms of the autophagic pathway, TFEB activation can lead to increased levels of autophagy. Overall, my project adopts a TFEB-based screening strategy to identify novel geroprotectors. To do that, we established a high-content screening platform to filter out drugs that can activate TFEB. We used cells that express TFEB with a green fluorescent protein attached to it. So this allows us to visualize where TFEB is in the cell. Essentially, what we want to see after drug treatment is the movement of the green TFEB from the cytoplasm into the nucleus. So this tells us that TFEB is activated and can act as a switch to turn on the genes in the clear network. Then, the identified TFEB activators were put through a secondary screen to confirm that this TFEB activation translates to an increase in autophagy activity. This time, we used cells that express LC3 with a green fluorescent protein attached to it. So remember how I said that LC3 is inserted onto these waste-containing vesicles during autophagy? Ideally, what you want to see after drug treatment is an increase in the number of LC3 green spots in the cytoplasm. So this would suggest that autophagy activity is increased. After putting 2,964 drugs from the FDA-approved and pharmacopoeia drug library through our two-step screen, we identified 43 drugs that can both activate TFEB and increase autophagy activity. Upon categorization of these 43 drugs, there was one group that particularly caught our interest, which was this class of drugs in dark blue, also known as the antipsychotic and antidepressant drugs. This group was interesting to us because autophagic dysfunction is implicated in both health span and lifespan regulation, and is also prevalent in psychotic conditions. Moreover, individuals with psychotic conditions exhibit accelerated aging. Hence, antipsychotics and antidepressants seem to play a potential role in modulating autophagy and lifespan. One example is lithium, which is used to treat bipolar disorders. It has found to increase autophagy and extend either health span or lifespan in yeast, worms, and flies. So following the same train of thought, we decided to first focus on drugs from the antipsychotic and antidepressant group and evaluate their potential as geroprotectors. We have yielded some promising preliminary results, which I hope to share with you guys in future. As we continue working on this project, I would like to thank the team for their efforts thus far. And I would also like to thank you for listening. Feel free to contact me if you have any questions.
Thank you, Grace. We are looking forward to hear more about the story of lithium and autophagy and how to increase that. But now we are going to Professor Felipe Court. He obtained his PhD at the Edinburgh University and focused on fundamental parameters regulating conduction velocity. He is now a full professor at the Universidad Mayor in Chile. And he is also calling in from Chile, where he established the Center for Integrative Biology. Currently, Felipe serves as the director and principal investigator of the Center for Neuroscience, Brain Health and Metabolism, also called GERO. And he is the chief scientific officer of Momentum Therapeutics, a US-based biotech biotech developing interventions for age-related conditions. We would really love to hear more also about that. Felipe, thank you so much of being here. You will talk about the central and peripheral innervation and its strategy to prevent and reverse age-related conditions. The floor is yours.
Okay, thank you very much, Andrea. I'm very happy to be here. In the lab, we are very good fans on these webinars. We have learned a lot about this. Then let me share my screens here. Good. Then, yeah, today I want to tell you what we have been doing in the lab regarding central and peripheral innervation, especially associated to age-related conditions, but also aging. And we started some time ago looking in the brain and in the nervous system. I am a neuroscientist, but now we are moving into the periphery to look organ innervation and how that changes in diseases, but most importantly, how it changes in aging, in kind of healthy, non-pathological aging. Then I will start with a couple of introductory slides for you to kind of get the whole idea of what we are studying. I will start with just telling you about axons. Okay, this is not a big mystery, but I want to just tell you some characteristics of this structure of this that are associated to neurons. As you probably know, axons have very, very big lengths in highly branched neurons, but also in long neurons. Here you can see reconstruction of axons in the mouse brain, but also in the human brain. And if you take kind of particular neurons, like a dopaminergic neuron that is associated to Parkinson's when they degenerate, and you sum up their axons and terminals, sum up 1 neuron, 0.7 meters. And if you go to another very kind of characteristic neuron, that is a human forebrain cholinergic neuron, the terminals can have a total length of 100 meters. And of course, we know that there's also not highly branched, but also very, very long neurons in our bodies. And in fact, the longest axons probably are in the blue whale that have 30-meter axons. This is a piece of cytoplasm that goes from one place to the body to another one. And in our bodies, the longest ones are 1.5 meters. I'm putting here in the corner PMIT references for you if you want to check some of this data. Then this is basically the basis of connection or connectivity between one part of the body with another in the brain, or between the brain and the body. And this is something that we have been studying, especially in terms of regeneration and degeneration. And something that happened along aging is that there's a massive elimination of axons and connections, not only in the brain, but also in peripheral organs. And that's something that we have been kind of interested for some time now and studying. In fact, this is important because loss of, for example, in the periphery, loss of somatic and autonomic innervation that have been studied mostly in adult organisms can lead to several changes in physiology. Of course, they decrease organ response, both in response to internal environmental stimuli. And as you can see here, this is an example getting rid of all the sympathetic nervous system of the pancreas and how the glucose tolerance goes down. Then basically, of course, if you eliminate innervation or this decreases, will have an impact in physiology. But also, the innervation increase inflammation and organ fibrosis. And something I have been studying in some particular tissues, it decreases also tissue regeneration. Then basically, non-pathological kind of age-related decrease in innervation in most of the organs that have been studied, and here are some examples, will lead to some or all of these kind of changes. Of course, in the brain, this also happened. There's not a massive loss of neurons like somas in the brain, but what's happened along aging is that we lose a good amount of axons, and here are the data that are quite impressive. We lost kind of axons in the white matter at around 10% per decade, and start pretty early, as you can see here. Then basically, loss of axons, not neurons, but just the compartment of a neuron seems to be important because it will lead to several changes, physiological changes in the body along aging. And this is something that we are interested and actively studying. This is part of my lab, basically, and we work in two things. And today I will tell you about molecular mechanism of axonal degeneration. Start by telling you that there's molecular mechanism, hence there's some genes and proteins that can be targeted to decrease axonal degeneration. And I will tell you some some data about axonal degeneration in aging and how kind of modulating that process could lead to improved function. Another part of the lab is related to regeneration, because it's another interesting topic. When you have loss of axon, you want to regenerate. And in fact, we have shown recently, and here is the link or the PMIT number, that there's an appearance with degeneration of axons of glial cell senescent that inhibit regeneration. Then there's also here some interesting targets for regeneration. Let me move here. Okay, then let me start about molecular mechanism of axonal degeneration. I will go through a little bit of story. This is the first study showing that axons degenerate when you cut them. Basically, when you separate axons from the soma, they will degenerate. And it was August Baller, a long time ago that described this. And then it took more than 150 years to realize that basically there was a mechanism associated to this process of axonal degeneration. Okay, this type of degeneration that is activated by severing the axon, basically separating the axon from the soma, is called now bilinear degeneration, and we have been using this in the lab to study a molecular mechanism that activates during the degeneration process. And here is how it looks, an intact axons in the sciatic nerve, and what's happened only 2 days after it's completely separating, mechanically separating from the soma. They degenerate very fast. They activate a program that I will tell you later how it's about. And of course, this model that allows us to assess defining mechanism of axonal degeneration. And something that is also super important is that resemble the mechanism activated in neurodegenerative conditions and neuropathies. Then basically we can use what we learn about this simple kind of stereotype mechanism of axonal degeneration that activates after damage to basically find some targets for neuroprotection. I will show you a couple of slides with the mechanisms that we have discovered so far. This kind of joint project, joint venture between several labs, of course. And what we know now is that axonal degeneration is independent from soma degeneration. Then basically the soma uses one mechanism to enter into cell death, that is mainly apoptosis, but the axon uses a completely different mechanism that could be activated by axotomy as bilinear degeneration, but also using other stimuli like chemotherapy, decreased axonal transport, and several other ones. These have been shown in several animal species, and what's happened briefly is that there's a decrease in a NAD synthesizing enzyme and an increase in a NADase that is called SARM1. This leads to decrease in NAD in the axonal compartment, leading to mitochondrial dysfunction and ultimately to axonal degeneration. More recently, what we have found is that necroptosis, the necroptosis pathway, okay, and I will tell you briefly later about this, is also activated and is probably a central part of the degenerative mechanism of these axons, okay? Especially when the stimuli is excitotoxicity or inflammation, but also take place after axotomy and chemotherapy. And what we have shown in the last year is that this process of necroptosis is involved in axonal degeneration in several conditions, including chemotherapy-induced axonal degeneration, glutamate excitotoxicity, and also a couple of diseases like Parkinson's and Alzheimer's. Okay. Then the key question here is the necroptosis pathway, that leads to axonal degeneration and is involved in axonal degeneration, is associated also to age-dependent axonal degeneration in the brain and in peripheral organs. And today I will show you data about the brain and hope to, in the future, to tell you some data about peripheral organs that we have been actively working also in this system. Okay, then let me give you necroptotic pathway in a nutshell. This is kind of the typical canonical way we see necrosis activation. Is activated by several kind of stimuli, including TNF-alpha, especially when caspase-8 is inhibited. Otherwise, TNF-alpha activate apoptosis. It's also activated by toll-like receptors, by viruses, LPS, and decreases NAD, something that we have shown. And then this activate RAP kinase 1 that is shared between necroptosis but also apoptosis. RAP kinase 1 phosphorylate RAP kinase 3 that in turn phosphorylate MLKL. MLKL is not a kinase, it's a pseudokinase that oligomerize and lead to membrane permeabilization, and leading ultimately to inflammation. That is basically the typical response of this necrosis, but this now regulated necrosis. Okay, this seems simple, but there's another kind of point that we have been aware to interpret the results, because there are several other things that take place. For example, RIP kinase 3 is also involved in inflammatory responses, independent from the full kind of necroptotic pathway that involves MLKL. Okay, and of course, this inflammation will feed back into the stimuli that activate necroptosis. Then things that we have to keep in mind. But also there's other pathways. Activation of MLKL can lead to mitochondrial changes that could lead to internal or subcellular ROS production. Then, of course, necroptosis, as you probably have heard, have been functionally associated to several organ aging and age-related conditions. And here is a list if you want to take a screenshot and then later look at the papers. But here you can see retinal diseases, liver aging, this testis aging and reproductive decay in males was probably the first paper showing in a basically aging without disease associated, and so on. What we start to think is that if axonal degeneration and loss of function was associated to a healthy brain aging. And let me jump into that and show you results from the lab. Then we start going into the brain of mouse to see if axonal degeneration was actually happening there, as has been shown in human brain samples. And for that, we look at the hippocampus, because it's a region with high connectivity that is known to be lost in aged humans, and there's some data in aged mice also. And also we have a very important function that is learning and memory that can be measured, but also is important for aging in the population. Then, as you can see here, when we look at axons, and these are the white kind of lines in the hippocampus, this is an adult animal of 6 months. When we look at aged animals, and this happened also in other brain regions, there's a very important decrease in neurofilament positive axons that we check using several other techniques and quantifications. And then basically axonal degeneration was actually happening. When we went to see necroptosis activation, okay, and remember here that here is the kind of the basic pathway of necroptosis, this is jumping, okay. Necroptosis activation, we saw actually that along aging, okay, RIP kinase 3 gets phosphorylated, as you can see here in red, but also MLKL gets phosphorylated and it gets activated along aging. And we saw this in different places, not all of the regions of the brain, but in some particular regions were much more activated. Then we move to see what was the consequences of this activation of necroptosis, and we first used a total MLKL knockout animal, and this is the NH animal, 24-month-old, that we compare with a wild type animal. We were kind of basically eliminated this step of necroptosis, and what we saw, using kind of regular techniques of behavior, is that the animal have better locomotor capacity, as you can see here, the MLKL knockout, the aged one, but also grip strength. And then we move to look a little bit on synapses and in the brain. This is in the hippocampus, and actually the MLKL knockout had better or more dense synapses compared to aged wild-type animals, and is less than the adult, but it's more comparable to these stages. And then, of course, we move to do functional tests, and I'm showing you here memory, learning and memory using the Morris water maze. Basically, this is a water maze where the animal have to find a hidden platform. And this is the learning curve for the animal in consecutive days to get better and better to find the platform. Then the latency goes down, especially in an adult wild-type animals in black here. That basically get worse when you look at aged wild types in blue. But MLKL knockout, the aged one, have learning performance that are comparable to the adult wild types animals, and of course adult MLKL knockout. Then basically these knockout animals have better capacity for learning. And when we test memory, a couple of days later, as you can see here, in a wild-type genotype, the animals, adults, are very good to find where the platform is located. That is actually located in this quadrant, as you can see here in the heat map. But the aged animals lose their capacity to find the platform. And the MLKL knockout, the aged one, is have a much better memory capacity, in this test. Then we look at axonal degeneration, and actually axons were looking much better in aged MLKL knockout in the hippocampus compared to wild types, and some other markers for degenerating axon that is shown here in green goes down in the MLK knockout, but also some activation of microglia gets reduced when we eliminate this MLKL gene. Then we move into a more kind of therapeutic or pharmacological approach, and what we did was given a very short inhibition of RIP kinase 3 using this drug, GSK, in very old animals. And we start with 23-month-old animal, and we gave only 1 month of the drug. This is by intraperitoneal implantation of an osmotic pump. And we look 1 month later. And what we find is that actually axons, after this very short-term treatment, looks better after RIP kinase 3 inhibition, as you can see here. And when we did memory tests, when we look at other parameters like inflammation, actually aged wild-type animals treated with the drug show a profile of pro-inflammatory cytokines that looks much like the adult animal rather than the aged wild-type animal treated with vehicle. And this was quite interesting. This is in the hippocampus, where we also did this in plasma samples with similar results. Then basically, it's not only in the brain, but also there's a systemic change in terms of inflammation that makes sense thinking on what are all these basically consequences of activation or inhibition in this situation of necroptosis. Then we move into memory testing, and the same experiment, of course. And as you can see here, we check the learning curve compared aged vehicle-treated animals with aged GSK-treated or RIP kinase 3 inhibitors. And as you can see here, the learning curve is much better, improves when we inhibit RIP kinase 3. This is systemic inhibition, as I showed you before. And also memory capacity, the capacity to find the place where the platform should be located improves. And this is not only with one RIP kinase 3 inhibitor. We have been testing several other ones. And let me show you here that if we use a different RIP kinase inhibitor, we have kind of very, very similar results, both in learning performance, but also in memory performance, as you can see here, down here. Then basically summing up, if we genetically or pharmacologically inhibit necroptosis, we can have decreases in axonal degeneration, decrease in inflammation, both systemic and in the hippocampus, like very region-specific, and these have impact in learning and memory performance, but also in some other parameters like locomotor and strength, rib strength, as I showed you in the previous slides. Then we did some very kind of unbiased proteomic studies in the hippocampus from wild type, adult, aged without MLKL, that basically MLK knockout, but also aged with rekinase 3 inhibition using GSK. And as you can see here, we profile a very good amount of protein. We use single shot in collaboration with Tom Wishart at Edinburgh University. And here is the profile when we compare age wild type versus adult wild type. Then basically this is the kind of the signature of aging in terms of proteins in the hippocampus of mice. And this is what's happened when we compare age MLKL knockout versus age wild type. Then there's a very important kind of reversion of some of the proteins that goes down, okay, that now they're going up when you knock down MLKL, and the inverse also is the same. Proteins that usually goes down with aging, are reverted with the genetic intervention. And the same happen when we inhibit rekinase 3, of course, in a different level, is a milder effect, but still is an important reversion of some signatures of aging. And when we look basically at canonical pathways, as you can see here, these are the ones that significantly changes in these two conditions, okay, genetic and pharmacological inhibition of necroptosis compared to the age signature. And here, as you can see here in synaptic mechanism that goes down with aging, they are reverted in these two interventions that blocks necroptosis. And there are other interesting like nutrient sensing, and basically, I want to point out synaptic LTP, for example, that get reversed, mTOR signaling, and senescent pathway that also, that goes up in aging, but is reverted by, by genetic or pharmacological interventions. Okay, and just to finish, I just want to show you some kind of validation of this data. We did senescence and other validation of the proteomics. And this is looking for LTP, okay, that gets somehow, as you can see here, reverted when we knock down MLKL or we use the rekinase 3 inhibitors. And we did LTP in the hippocampus in collaboration with Waldo Serpa at Catholic University here in Chile, when we induce LTP by high-frequency stimulation, and we look the response. And as you can see here, okay, in the adult, this is basically LTP in the adult wild type in black, and that goes down in aged wild type. But when we knock down MLKL and we look at aged kind of MLKL in green, we have a reversion or kind of normalization of LTP in this genetic intervention. And in some extent, but significantly, we also get a reversion, as you can see here, in the kind of cyan bluish line when we treat the animals with GSK that is comparable with the adult wild type LTP performance. Okay, and with this, I just want to point out that maybe RIG kinase 3 inhibition could be a safe target. Something very, very interesting about RIG kinase 3 and MLKL also is that have been lost several times in evolution, okay, in several lineages, okay, and in a lot of species is of course not required for living because it's not there or it's not functional. And let me show you some examples here. Of course, in primates is present, okay. Then we have, of course, RIG kinase 3 and MLKL. But when we look, for example, in rodents, there are some species like rat and mouse that have RIG kinase 3, but several other ones like the naked rat, mole, and degu that do not have RIG kinase 3 or is not functional. Also in Afroteria, okay, some species have it, some other don't have it. As you can see here, the red one is the one that RIG kinase 3 is not there or is not functional. And all cetaceans do not have a functional RIG kinase 3. Okay, I'm not implying, of course, that there's a functional association between not having RIG kinase 3 and longevity, but this is an interesting thing to consider and, of course, to study. And having said that, let me finish with a couple of things that we are doing now. We are dissecting at the moment the specific role of RIG kinase 3 in Parkinson's disease using conditional RIG kinase 3 knockouts, a novel compound that can enter or not to the brain. And this is a collaboration or the second project with the Michael J. Fox Foundation. Then we are targeting or eliminating RIG kinase 3 only in neurons or immune cells to start with, to see the contribution of RIG kinase 3 pathway in these different populations. What we have been doing, and we are close to getting out, is showing that RIG kinase 3 inhibition can rescue the degeneration of axons in the periphery after chemotherapy-induced neuropathic pain, both in adult and aged animals. And finally, we have been moving to other conditions that are not associated specifically to the nervous system. And we have recently showed that in age-related ventilatory impairment, that RIG kinase 3 knockouts, mice have chemoreflex responses that is very kind of important for the aging process that are indistinguishable from adult wild type. And here is the publication. And with that, I want to finish by, of course, thanking all the people from the lab. Okay, most of the data about necroptosis in brain aging was done by a former postdoc, Macarena Razola, that just moved to open his lab here in Chile, but also a lot of people in the lab, and of course collaborators that are named and funding bodies that have been, of course, super important to perform all this work. And with that, I will be happy to discuss this data and take questions. Thank you very much.
Thank you so much, Felipe. What a wonderful presentation for me, a little bit new topic, and maybe also for others. Let me start quite easy. How, if you look at different animal models, think about C. elegans, the mice and the rat, how different are the nervous systems with the human ones? And if you would be able to choose one of the animal models, so not only thinking about C. elegans and the zebrafish, etc., what we always study, what would you study to answer your questions?
Okay, yeah, that's interesting because the selection is is more associated to what you want to do rather than the specific molecular mechanism. Something that is important to know is that at least the pathway that is associated to NMNAT, decrease. Basically, I went very fast through that because there's a lot of information there, a lot of research on that. But basically what is happening in the pathway is that NMNAT, that is critical for NAD production, goes down in the axon. And, SARM1, that is a metabolic sensor that senses NAD kind of levels, get activated when NAD goes down. Then it activates NADase activity and start to consume NAD. Then NAD goes super down in the axon. those proteins are pretty well conserved in different species, okay? Then if you want to study the core kind of axonal degeneration mechanism, you can actually use the different species and choose what is best because you want a specific readout, you want to have more genetics, etc., etc. Then we have used actually C. elegans to study axonal degeneration, okay? Because we can look specific neurons and look the whole process, in vivo, and that's a fantastic model. And you can do genetics, you can target neurons or other cells. And basically, that's a very, very good model. For example, if you move into Drosophila, the main problem of Drosophila is that they don't have the necroptotic pathway. Then basically, you know that Drosophila, a lot of things have been reduced or are reduced to kind of single proteins. And in terms of kind of apoptosis and necroptosis, it seems that at least we haven't been able to find a proper necroptotic pathway. They have axonal degeneration that is NMNAT, SARM1 dependent, not dependent, but it seems to be not associated to necroptosis activation. Now, zebrafish, of course, have kinase 3. There's some kind of debate about MLKL, but also it's a very, very good model. Then, answering your question, you can use all this model. Of course, we are starting a project with zebrafish, or we want to start kind of trying to find out if MLKL is there and is functional, but we want to start a project in zebrafish, but we have been working a lot with basically mouse, because also we can check for just some kind of behavioral readout that we are interested, especially learning and memory that we have been very interested. I'm not saying that you cannot check that in the other models, but we are more expert on that. But there are several models that you can use, and information about the mechanism of axonal degeneration have come from different models. It's basically the pathway I showed there is a contribution from several groups using different animal models.
Yeah, so it's always about what mechanism and what's feasible to do. So, and of course, C. elegans is quite feasible, as you said. I very often ask the researchers being on this show, what is the real contribution of your pathway you are studying to the aging process? We have so many different mechanisms, etc. And there are lots of listeners also who want to find their mechanism and want to be guided. So tell us a little bit about how much, if you would have no necroptosis anymore and you would really nail it, how much of the aging process you could stop?
With you mean by just targeting necroptosis? Yeah. Well, I think when we are used to look at the hallmarks of aging at the start, like a fixed and separated entities, and now, of course, these have evolved much more, and we are starting to integrate, the hallmarks, and we have found out that when we start to integrate, everything or most of the things get kind of associated, right? We have like a super dense network of kind of a connection between the hallmark of aging. I think one of the hallmarks that are necroptosis are probably very, very is associated is, of course, inflammation. I think one of the basic kind of roots of inflammation probably is activation of necroptosis, especially because you have this feedback loop that basically necroptotic activation by pathogens, by viruses, okay, will lead to inflammation, okay, that will be resolved, okay, in a young or in an adult organism. But in an aged organism, this resolution of inflammation will not be as kind of efficient as in a younger organism, and that will probably further activate necroptosis. necroptosis. Then it will create a kind of a feedback loop that will increases. And we and others have been looking for different tissues. And actually, necroptosis is activated in most of the tissues with aging. Okay, then basically, inflammation will probably be the kind of pathway that, or the hallmarks of aging that will be kind of contributed a lot by necroptosis activation. I don't know if.
Yeah, inflammation is there with the root cause. So if we tackle that, you're jobless. Is that right?
Yeah. Well,
okay.
I
don't think that's
we do a lot of more experiments on necroptosis, and we have been trying to find out, for example, molecular mechanism that leads to kind of long-term necroptosis activation by a single stimuli. Okay, there's something interesting about necroptosis that we don't understand, and we are trying to kind of to get into kind of actual data, right?
Yeah, no, absolutely. I think since Claudio Franceschi termed that that key term, I think it's we didn't solve it yet. So please continue with your great research. RIP kinase 3 inhibitors, will that be a new drug for dementia?
Well, at least what we have been getting from animal studies, RIP kinase 3 inhibitors are very, very good for inhibiting basically what I will better say my cognitive impairment or cognitive impairment. We haven't tested kind of dementia itself. What we have done is test animal models of Alzheimer's, and it works pretty well in, with cautious, because animal models of Alzheimer's are having, we have to take the result with cautious. What we have basically seen is that it improves basically kind of age-related decrease in memory performance. Okay, I will say that to be cautious on that. The good thing that we have been finding is that RIP kinase 3 inhibitors are very well tolerated, okay, systemically, and we have done tox studies, and also we have been trying, and this is our kind of future idea, trying to find compounds that get into the brain, okay, and there's some of them that are very good to get into the brain, and some of them that stay in the periphery, and that could tell you that you can target at least pharmacologically different regions. I know that I didn't answer your question. I think we need more studies for that.
I absolutely understand. I think if you nail to find a drug against dementia, you are absolutely in there. But before going to Sanjay Kumar, our research fellow at the Healthy Longevity Program, I would like to ask you one last question with a short answer. You set up a wonderful anti-aging or a program in Chile. Any learnings from others who would like to establish a new center for aging or health and longevity in a country?
Yeah, well, yeah, I mean, we started this about 9 years ago. We have a lot of help from the Back Institute, especially Brian Kennedy, when he was president there. He was involved a lot in generating this model, okay, helping us because we have no experience. I never worked in aging. I was a neuroscientist working in diseases. And at that point, we really wanted to move on to that. And then basically we gain a lot of experience. We are working with human courts. We are establishing the first aging courts here in Chile, very small because funding, but we are, basically setting up kind of a mechanism for protocols to do court studies in South America, in Chile especially, and then also moving several kind of group of research from diseases to aging. And I think this is having very kind of important for us, and is something instrumental was to basically collaborate with scientists that were doing this abroad. And as I told you, they have helped us a lot, saying this is disease, this is aging, okay? You have to move into one.
Even there's lots of discussion in the field, but I know that there are lots of questions out of the audience. So please, Sanjay, please take over and continue.
Thank you, Prof Andrea, and thank you, Prof Court, for enlightening the role of central and peripheral innervation as a strategy to prevent reverse age-related conditions. So I'm going to ask few questions from audience. So is outside the hippocampus or memory effect, do you expect that these inhibitor would also have beneficial effect with respect to axonal recovery from spinal cord injury or degeneration?
Yeah, well, there's some studies showing that necroptosis could have some effect over traumatic brain injury, TBI. Then basically it's a mechanical injury that you can think that could have some parallels to other injuries like mechanical injuries. In fact, what's happened in this type of injuries is that basically the axons that don't get directly damaged, those will degenerate. Basically, there's no way that they reconnect, at least in mammals. But those that are not damaged, but they're very close to a kind of pro-inflammatory milieu, basically they get pro-inflammatory molecules, they will activate necroptosis. And basically we have shown that activation, or we and others have shown that inflammatory kind of cytokines and other inflammatory molecules activate necroptosis and degenerate axons. Then basically, in principle, okay, you will not save the axons that are damaged, but a lot of axonal degeneration in this condition, for example, spinal cord injury, are not the ones that are directly damaged, but the ones that are degenerated later because you have inflammation, but also because you have a glial cell death that also, can be in principle protected by kind of inhibiting the axonal mechanism for degeneration. Yeah, that's kind of a guess, but is actually based on some data on TBI. Okay.
Thank you, Professor. So my next question would be, so necroptosis and apoptosis are closely linked and feedback on each other. Would you say there is a more proportion of apoptosis or necroptosis in aging?
Good question. Yeah, I think it depends on the system. We have explored apoptosis in different brain regions. In general, with aging, healthy aging, there's very few neuronal apoptosis in the hippocampus. Okay, there's but there's a small contribution, I think, compared to the amount of apoptosis that is there. Okay, of course, this changes from tissue to tissue. Something that is important, I think, is critical for to kind of move a little bit from your question, is that we have done experiment where in the same neuron, okay, we can basically activate the axonal compartment or the soma compartment. That in vitro you have to do microfluidics, for example. In vivo, there's a physical separation, and while you can activate a necroptosis in the axonal compartment without activating apoptosis in the soma compartment, and the other way around is not possible, because when you activate apoptosis in the soma blasts, and it's kind of an axotomy, and the axon gets into necroptosis. But what I'm trying to say is that, the neurons have two different mechanisms of degeneration in the same cell, okay, but in different compartments. And something that is interesting about necroptosis, at least in diseases, this haven't been studied in kind of aging, is that when you have a necroptosis or axonal degeneration, okay, this will lead eventually to apoptosis of the soma. And this have been studied in Parkinson's, okay, and other diseases, where you have like a dying back degeneration, where terminals start to degenerate, for example, in the striatum, and then axonal degeneration kind of progress, and later on, okay, you have apoptosis of the soma, okay? Then this is loss of the soma is a kind of later event.
Thank you, Professor. So my next question would be is there any RIP kinase 3 or MLK3 specific target molecules in clinical trials that are associated with neurodegenerative diseases?
Yeah, no, at the moment, what have been advanced is RIP kinase 1 inhibitors, in fact, and there are some RIP kinase 1 inhibitors in clinical trials for more inflammatory conditions. But still there's I think RIP kinase 3 and MLKL that have been a lag on basically moving this into the clinics compared to RIP kinase 1. Okay.
Thank you. And so I'll combine one or two questions so that maybe it's more clear. So there are few members of certain family that are reported. They are associated with the peripheral neuropathy, and there are the NAD metabolisms that are also very reported in the peripheral neuropathy. So are you think boosting of NAD with some boosters can be a potential candidate for this to cure peripheral neuropathy or these kind of diseases?
Yeah, that's a very interesting question. There's a lot of things there. I will be kind of trying to answer. Have a short answer on that. Yeah, what we have found, at least in vitro conditions, is that NAT decreases is very, very associated to activation on axonal degeneration. And we have been found, of course, that if you boost NAT, but in vitro condition, and we have some unpublished data in vivo on peripheral neuropathies, in fact, you can actually reduce necroptosis, okay, in peripheral axons and reduce axonal degeneration and loss of function. Then basically the answer is yes, probably, boosting NAT, okay, will in some way decrease necroptosis, and that will lead to decrease in axonal degeneration and functional kind of recovery or decrease in functional loss. Yeah, that's a short answer for a lot of data is going around there.
Thanks, Professor. So another question is most of the neurodegenerative diseases or other diseases are associated with protein misfolding and amyloid-like deposits. So what do you think, so necroptosis associated with amyloid formation in the brain and other organs as well, or just is specific to the neurodegenerative diseases only?
No, I think, necroptosis is associated to several other kind of changes, intracellular changes, and that could be, inflammatory or kind of changes in mitochondrial dysfunction. Then there's some data showing that reactive oxygen species can actually directly activate necroptosis. Okay, also, increasing calcium by TAMP kinase 2 activation can also activate necroptosis. Then basically, this is not restricted to a single pathway. Then in the axonal or neuronal kind of in the neuron, okay, protein misfolding usually leads to a decrease in transport, axonal transport, and decreasing axonal transport actually will activate axonal degeneration by different mechanisms.
So my last question would be what can we do to improve or slow down our axonal degeneration?
Well, yeah, you can take RIP kinase 3 inhibitors when they're, up there in the clinic. But no, now I think I will say that we need some more data about NAT supplementation, okay? But this is at least looking the in vitro data, okay, that they are in the moment, and there's some data close to get out. Probably that could be some way to decrease axonal degeneration. But there's other ways that is decreasing necroptosis. Of course, if you decrease, inflammation, inflammation by different ways, you decrease axonal necroptosis activation, and we are actually doing some interventions in animals, looking for kind of more lifestyle changes that decrease inflammation, and we have been seeing very, very interesting data that they actually decrease necroptosis and axonal degeneration. And there's some data, and there's one paper that is out there for some time, and it is part of the one that I cite there, that also dietary restriction, is also associated to a decrease in peripheral necroptosis activation. And basically, there are some ways to decrease necroptosis activation that will lead to a decrease in axonal degeneration, probably, that they have to be studied, but there's some kind of interesting data out there.
Okay, so I'm hand over to Professor Andrea. So,
yes, thank you so much. Thank you, Felipe. Thank you, Sanjay, for this excellent Q&A. I'm looking forward until we have the RIP kinase inhibitors in clinical practice. So hopefully we will solve lots of problems we are seeing here in clinical practice. Thank you so much.
Yes, thank you for this invitation. Was been super interesting to talk and discuss about this.
Thank you. And thank you, our listeners, for being with us. Please use the chat function to leave comments and your feedback. It's highly valuable to improve even this session. I have a couple of announcements. Join our Unlock Healthy Longevity Supplements Conference. Yes, it exists. From the 29th of February to the 1st of March 2024. And this is the first global scientific conference solely focused on the role of supplements as a geroprotective intervention. And Singapore will be the epicenter of leading scientists, clinicians, industry partners, government representative, consumers to explore what kind of supplements we should take. Or maybe none? I think a couple. The NUS Medicine also recently launched the Academy for Healthy Longevity. Please sign up for the 3-day intensive course, and that's happening at the 26th to the 29th of February 2024. It's a 3-day super intensive conversation with scientific and clinical leaders, followed by the supplement conference. The places are limited, half are already being filled, so please sign up to secure your spot. Another highlight is the Healthy Longevity Talent Incubator in June next year for postgraduate students and early career professionals. Also, that's now being open for admissions. The next episode will be on the 16th of November. I will host, that should be December. I will host Dr. Yanni van Lochem. He is an aesthetic physician and he is the medical director of the UMA Institute here in Amsterdam. Our final show, we always end with a video, and this time it's about Queen Lily. Take care.
She was 22 years old, which already is exceptionally high. There was a lot going on in the run-up to her turning 23, which in cat years is 108 years old. The journey started way back in 1999. As she's gotten older, I've noticed that she is much more dependent on me. When she had a stroke a few years ago and I was seeing how she was walking, I thought, this is it, she's not got long left. When I'm at home, I appreciate the time that I have with her because I know that nothing is endless. But she pushed through, she's just defied the odds. She still has the strength to jump up very high. She's still going, still strong, still full of life. She loves her life and just is determined to live it out as long as she can. She doesn't hold back. A wonderful birthday, and then slept like a baby. Lily has shown me the true meaning of what it is to be a soulmate. Someone that is literally at your side till death do you part. she's been the best friend I could have ever asked for.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.